To complete plant sampling, you will need the following items: a falling plate meter, a 16 by 16 inch PVC quadrat, scissors, small sampling bags for plant DNA. You'll need one small paper bag per transect and one plastic gallon Ziploc for all transect bags per site. You'll also need silica beads. All sampling processes will occur at the same time as you move through the transects. The sampling order at each location along the transect should be: first, the falling plate meter, the green cover, total cover, and species richness. Plants will then be collected for later identification. Forage analysis will be the last sampling method.
Now we're going to do forage.
First, we'll sample plant biomass using the falling plate meter. To construct your own falling plate meter, refer to the link in the caption. The falling plate meter is used at every transect. Readings will be taken starting at 0 meters and continue every 2 meters along the transect line until 50 meters. Place the tip of the metal rod on the ground surface of the area to be measured. Gently let the plate fall to the surface of the forage or crop canopy.
You know, if you measure your...
Read the number on the pole where the pole and the PVC pipe align. Record the measurement in whole or 0.5 centimeter increments. If the ground is bare soil, record 0 even if the meter gives a small reading. Avoid setting the drop plate on sticks and lumpy soil that may throw off the readings. Continue every 2 meters until all the readings are complete. To estimate green and total cover, we'll use the quadrat. Each measurement will be correlated with the falling plate meter, occurring once at the 0 meter, 25 meter, and 50 meter mark. At these meter locations, place the quadrat directly over where the falling plate meter was placed. To determine green cover, visually estimate the amount of green biomass covering the soil within the quadrat. Count crops as a part of the green cover. Record green cover percentage on the datasheet. Estimate the total cover, both brown and green biomass, protecting the soil surface within the quadrat. Count dead leaves and sticks. Record total cover percentage on the datasheet. Once cover measurements are recorded, determine the plant species richness in the same quadrat. Take a small sample of each plant species. Collect a leaf if possible, not just a seed head or stem. The sample should be a minimum of about 10 grams once dried, about the size you see here. Count the number of individual plant species. Record the total number of species on the datasheet. Place the samples in a small paper bag, using a different paper bag for each transect. Place all 4 paper bags into a gallon Ziploc bag. Add a cup of silica beads to the gallon bag. Make sure the plastic bag is sealed and the beads can flow freely through the bag. After 24 to 48 hours, check the silica beads. If they are orange, remove the silica beads from the gallon bag containing individual samples. If they're green, the beads should be replenished so that drying can be completed. Forage samples are collected at the 25-meter mark of each transect. Place the quadrat directly over where the falling plate was placed. Separate foliage so that the frame lies on the ground. Cut forage close to the ground, collecting all green material. Separate clipped material into 2 equal amounts that will each yield roughly 10 grams of dried material, about what you see here. Collect additional plant material from just outside the quadrat if necessary. Collect only green-growing plants if possible. Do not collect sticks, fruit, soil, roots, or dead plant material. Place the clipped material into 2 large paper bags. Leave forage bags out to dry prior to submission so they don't get moldy. And that's all for plant sampling. Good luck with the rest of your data collection.